Welcome back. I just wanted to clarify or emphasize something from our previous section that I don't think I gave as much attention as I'd like to. The cholesterol in our diets, all of it, 100% comes from animal foods. So those of us that are practicing a vegan diet don't have any dietary cholesterol. This is not a problem for them because all of us can make cholesterol in our livers. So they're able to make their own cholesterol, but they're not consuming any dietary cholesterol. So I just want to make sure that part was clear. This section is our last section of Chapter 7, and we are going to focus on lipid metabolism and things like lipoproteins. So here we have an example of our lipid digestion and how important bile is for that process. So dietary fat, mostly triglycerides. So that's kind of what we're focusing here on the digestion piece. As mentioned before, we do have enzymes in the mouth and in the stomach. So in the mouth, you have your lingual lipase. In the stomach, you have your gastric lipase. But for the most part, not a lot of actual chemical breakdown of the fat molecules occurs in the mouth or in the stomach. The majority of that is going to occur in the small intestine. One of the reasons is that we need this cool guy called bile to help us with that process. So bile is manufactured in the liver, it is stored in the gallbladder, and then the gallbladder is able to shoot that bile into the small intestine where it can go do its magic. So in the small intestine, the food that came from the stomach is going to be slowly added into the small intestine. We are going to have bile coming in. We are also going to have other pancreatic juices coming in. So we've got digestive enzymes as well as bicarbonate coming in from the pancreas. And that bile is going to take this fat, all of the dietary fat that we just ate, which really doesn't want to hang out with all of the gastric juices and the pancreatic juices and everything else. It doesn't want to hang out because it's nonpolar, and all of those substances are polar. They're water, very watery based. So it wants to get far, far away from them. So if it had its way, it would just continue floating on the top of everything. So we need to get these things together. We need to be able to surround fat into smaller little manageable pieces or chunks. And the way that we have that happen is actually through bile, the effects of bile. So for example, if you were to just pour oil into a cup of water, that oil is going to sit on top of the cup of water. However, if you shook it up, you'd be able to suspend little tiny droplets of fat within the water, but you'll see that very quickly those fat droplets will want to separate out. They'll want to float back on top of the water. If, however, we add an emulsifier, something like lecithin or something like bile salt, then we can keep those fat globules in suspension. And this is how we make salad dressings. So if you take oil and vinegar and just shake them up, you can very quickly pour that on your salad and eat it right away and it'll be fine. But if you wanted to make a salad dressing and then stick it in the fridge for later, if you didn't want to have to shake it vigorously every time you wanted to use that salad dressing and you wanted those little particles of fat to stay nice and suspended throughout the solution, what you would need to do is add something that emulsifies. And the way that emulsification works is that you have a compound like lecithin, like bile salt, where part of the molecule likes fat and part of the molecule likes water. So with bile salts, you can see that the water soluble part is over here. The lipid soluble part is over here. So it looks very much like a phospholipid in that sense, because phospholipids, the head of the molecule, likes water. It's the phosphate group. And then the fatty acid tails hate water. They want to get away from it. And so these portions are going to orient themselves around the fat globules in such a way that you are able to keep water hanging out with water and fat hanging out with fat. The fat doesn't feel so overly threatened by the water that it has to get away. It's able to stay suspended in these little droplets. So this is mostly happening in the small intestine because that's where bile is secreted. So interesting fact about bile is that it is both made out of cholesterol as well as out of heme. And if you'll remember heme came from red blood cells. So as those red blood cells reach the end of their lifespan, they live for about 3 months. So as that cell is being broken down, the heme portion is able to be recycled and used to make bile. So kind of neat. So now that we've got everything in these smaller portions, we have a better surface area to volume ratio. That is when we can finally utilize those enzymes, that lipase. So we've gotten some pancreatic lipase to join the party now. That lipase is able to take and separate those fatty acids off from the glycerol molecule in a triglyceride. The end product, when we are complete with that digestion process, that chemical digestion process, is going to be a monoglyceride. So it's glycerol and one fatty acid still bound together, still esterified there. However, you also have two non-esterified fatty acids. So non-esterified meaning it doesn't have that ester bond. It's not bound to the glycerol molecule any longer. So monoglycerides, non-esterified fatty acids, and bile acids form this compound called a micelle. That micelle is what gets absorbed into the enterocyte, remembering that the enterocyte is the small intestinal cell. So lipids are not water soluble. They have to be packaged inside of something else in order to be transported in the body. And we will go over several lipoproteins. We'll talk about chylomicrons. We'll talk about very low density lipoproteins, low density lipoproteins, and high density lipoproteins. You may better know these things as VLDL, LDL, and HDL. But we'll talk about those. The lipoprotein that we use for absorption and transport of fats, dietary fats, is going to be the chylomicron. So we've got our chyme, right? Chylo. Maybe that's a way to remember it because it's coming from digestion. It's from the postprandial, what we've just eaten. So it's the lipoprotein that's transporting all of the fats from our postprandial setting. The majority of what's inside of a chylomicron is going to be triglyceride because, again, that's mostly what we're eating. The lipoprotein itself has phospholipids in it. It also has proteins in it. Therefore, it's a lipoprotein. There are various different kinds of lipoproteins, and the different parts of the body recognize different lipoproteins and kind of let them in when they see them. So it is going to take that newly absorbed triglyceride and it's going to take it to what we call extra hepatic tissues. Extra meaning outside of, hepatic, hepats the liver. So extra hepatic tissues. So looking at another example of a lipoprotein, this one is LDL, you can see kind of the basic structure of a lipoprotein. So we have phospholipids. We see that the water-loving head is oriented to the outside. We see that the fat-loving tails are oriented to the inside, and that makes sense because we're trying to get these fats to hang out in a watery environment. So we want the watery part to be on the outside, and then we want the fat part on the inside because these fatty acid tails have no problem hanging out with the triglycerides and the cholesterol and all the other stuff that's inside of here. So when you think of lipoproteins, think of them as transport carriers, like little buses driving around different kinds of lipids, driving around different kinds of fats. So the chylomicron likes to drive around triglycerides. Our LDL is going to drive around different stuff. So we've got each of them has their own job to do. And really just kind of looking at the chemical structure, you see that it's mostly phospholipids on the outside, actually making up the membrane of the phospholipid. We do have proteins embedded in there. So that's a protein, that's a protein, that's a protein. The type of protein gets a special name depending on what kind of lipoprotein you're talking about. So with LDL, it was that apolipoprotein 100. Now, chylomicrons do have to travel through the lymphatic system first. So once that chylomicron travels through the lymphatic system, then it will eventually, the lymph system catches up to the circulatory system and kind of dumps everything in there. So once it gets in there, we have to make sure that it is going to do well in a watery environment. And that's why it's being transported within a lipoprotein. So Extrahepatic, as I mentioned, just means outside the liver. So any cell that's not the liver is going to receive what we just took in our diet. So chylomicron mostly has triglycerides in it. And those triglycerides are things that the body tissues want. So fat cells want to absorb fat and save it for a rainy day. Muscles want to absorb fat and use them for exercise, use them to fuel exercise. The heart is a muscle, and the heart muscle uses a lot of fat. That's actually its primary source of energy. So we need to deliver these to all the right places in the body. The chylomicron goes towards the cell. The cell has this enzyme called LPL, lipoprotein lipase. That lipoprotein lipase is going to allow what's inside of the chylomicron, which is triglyceride, allow it to break that triglyceride into monoglycerides and fatty acids. So it is a little bit complicated in terms of fat digestion, that we break things down in order to absorb them, but then we build them back up again, put them in a chylomicron as full triglycerides, send them across the body only to have them broken down again. Seems like a wasted step, but that's the cost of doing business. You really can't get in or out until you make it as simple as possible. So that absorption required it being in its simplest form. But during transport, it gets re-esterified and turned into a triglyceride again. And now that we're delivering it to the cells of the body are going to break it down again using that lipoprotein lipase that they have there. So that's the enzyme that the cells have in order to make sure that they can use the triglyceride and break it down into monoglyceride and fatty acids. So as I mentioned, it's either going to be stored for later or we're going to use it for energy. Another way to think of extrahepatic would be to call it peripheral tissues. So hopefully that makes sense. Please let me know if you have questions. It's not an easy process. It's a slightly complicated process. Your book does a pretty good job at looking at it, but there are other resources I could always point you to as well. So what we have left over after we've dumped those triglycerides off and delivered them where they need to go is this thing called a chylomicron remnant. So it was a chylomicron when it was all full of triglycerides. Now that it's done its job, it's delivered all those triglycerides to those extrahepatic tissues. Now that chylomicron remnant needs to go back to the liver so the liver can say what it's going to do next. So it will travel to the liver, it'll get recycled, broken down, and we're going to package some of those broken down pieces into another kind of lipoprotein called VLDL, so very low density lipoprotein. So very low density lipoprotein also transports triglycerides, but VLDL is going to transport triglycerides that we made ourselves in the liver. What are the two fatty acids we can't make? Omega-3 and omega-6. So those are only going to come from the chylomicron from dietary intake. But the liver's great at making a lot of other kinds of long chain fatty acids. And so it can package those up into triglycerides, put them inside of a transport vehicle called VLDL, and send them to other places in the body. So the job of VLDL is to take fats that we made in the liver and deliver those to extrahepatic or peripheral tissues. All right, so chylomicron remnant goes to the liver breaks down the chylomicron remnant, creates this new lipoprotein called VLDL, and inside that VLDL, it sticks a bunch of the triglycerides that it made. So once it has that, it will send it off on its way to go do its job. VLDL is different from chylomicrons in that it also has some cholesterol that the liver made. So it's not only got those triglycerides, but it also has a higher proportion of cholesterol than the chylomicron had. Chylomicron has some. There is some amount of dietary cholesterol unless you're eating a completely vegan diet, but it's not a lot. It's not a very large proportion of what the chylomicron has in it. Whereas the VLDL has a decent amount of cholesterol in it. So it's going to take those triglycerides to fat tissue, adipose tissue, and muscle tissue, and it's going to deliver it so that that can be either used for energy, we can break it down and turn it into ATP, or they can re-esterify it and store it as fat. So it can take those fatty acids and glycerol pieces and turn them back into triglyceride, and the fat cells can store them for later. So it is what we are going to use to deliver the fats that we made, as well as some cholesterol that we made. And then we have this intermediate density lipoprotein that is going to be the remnant or the leftover of VLDL. So as I mentioned before, we have this cool LPL, lipoprotein lipase, and that's how each cell is able to access the fats that are in there that are being delivered. Once VLDL does its job, the leftover piece is called intermediate density lipoprotein. So intermediate density lipoprotein, to give it a little bit more detail, taken up by the liver. So once the VLDL did its job, we have this leftover piece, just like we did with the chylomicron remnant. The intermediate density lipoprotein has to go back to the liver and get repurposed to do the next things that it might need to do. So the liver is going to say, okay, well, we're going to take cholesterol, we're going to turn it into bile. We are going to take this remnant of this lipoprotein, and now it's no longer VLDL. Now we're going to call it LDL. So low density lipoprotein. It doesn't have a lot of density. It floats pretty well comparatively, and it compared to like HDL. So what LDL does is it's going to deliver cholesterol. That's its primary product, right? That it's selling to all the cells around the body. It's delivering cholesterol. So LDL is highly regulated by the liver. And by the amount of cholesterol being made in the liver. So if our liver is making a lot of cholesterol, it's going to make a lot of LDL, and that LDL is going to go out and drop cholesterol off at these various recipient cells in those extrahepatic tissues within the body. A diet very high in saturated fat is going to make it more likely that you have a lot of LDL because you have a lot of cholesterol. We also have this other lipoprotein that the liver is going to make, and that is called high density lipoprotein. This high density lipoprotein actually does the opposite. What high density lipoprotein does is it goes around the cells of the body and various parts of the body, and it takes leftover cholesterol and it brings it back to the liver so that it doesn't just clog up the pipes. So HDL is kind of your cleanup crew. It'll get rid of cholesterol that maybe got deposited where it didn't belong, and it can start bringing it back to the liver for recycling. So higher, you're always going to have more LDL than HDL, but too high of LDL is bad for your health because it indicates that you're more likely to have cholesterol building up in your blood vessels and creating atherosclerosis, narrowing the arteries, hardening the arteries, and creating a higher likelihood for things like stroke and heart disease and heart attack. Versus LDL or HDL, for example, you want to try and bring that number up, and it will take some of that cholesterol back to the liver, get it out of the way so it's not clogging up the pipes, and hopefully create some better health outcomes for you. At the end of the day, we're able to use things like cholesterol to make bile, to make hormones, to make vitamin D. So the liver wants to be able to have some cholesterol to do those things. So intermediate density lipoprotein, as I mentioned before, is that leftover piece from VLDL, returns to the liver. We're able to package that into some LDL, low density lipoproteins. Those have primarily cholesterol inside of them. So we have cholesterol receptors on cells, and the cell will say when it wants some cholesterol, it's going to take some out of that LDL. And then eventually we're also going to have HDL that's made here in the liver that's going to actually take some cholesterol when we have too much cholesterol at places we don't want it to be, and actually return it back to the liver. And it can use that cholesterol to make bile. It can use that cholesterol to make vitamin D, testosterone, estrogen, all that cool stuff. I will also embed this video in your supplemental videos so you can see a little bit more about LDL, HDL, and hopefully understand that process a little bit better, because I think videos are great for explaining complex processes. So here's a little summary slide. Maybe the other slides, you want to kind of tie them all together into one. So in the intestine, we're eating, chewing and digesting mechanically and chemically some lipids. Eventually, we absorb those lipids. They have to be transported inside of the lipoprotein called a chylomicron. Chylomicron is going to deliver triglycerides to various parts of the body, the peripheral tissues, those extrahepatic tissues. We have some leftovers. Not everything gets used up, and certainly the lipoprotein doesn't get used up. So anything left over after delivery gets to return to the liver. We're going to have some triglycerides that are made in the liver, as well as some cholesterol that's made in the liver. Majority of what's inside of a VLDL is mostly going to be triglycerides, but a little bit of cholesterol. Those very low density lipoproteins will deliver some triglycerides. That's their job, is to deliver some triglycerides to the body. There are going to be some leftover pieces. Those leftover pieces, we're going to call them LDL. So those LDLs, those low density lipoproteins, now that we've delivered out that triglyceride, what's left over is the cholesterol, and that's what changes the density, is the ratio between triglycerides and things like cholesterol, sterols. So those LDLs are going to deliver cholesterol. Anything left over comes back to the liver. Thankfully, the liver also has these HDLs. The HDLs are going to actually come pick up some cholesterol, bring it back to the liver for some recycling. So really high LDL is more likely that you're going to have cholesterol adding itself to places it doesn't belong. Higher HDL, trying to increase the amount of HDL you have, hopefully means you've got a better cleanup crew. You can get some of that excess cholesterol put where it needs to go, brought back to the liver for recycling, so it can make things like bile and vitamin D and testosterone and estrogen and all that good stuff. So lipoprotein composition changes, chylomicrons, mostly triglycerides. In blue, you can see that there. A little bit of cholesterol. We've got some various different proteins in there, as well as some phospholipids. Phospholipids are purple. We've got our proteins, which are pink, but the majority of what's in a chylomicron is triglyceride, and that's why it's nice and fluffy. It's a lower density than even the very low density lipoprotein. I wish that they named them better to make it less confusing for you guys, but it's less dense than even the very low density lipoprotein. If you look at the VLDLs, you can see, they're about 60% triglyceride. We're making those triglycerides in the liver. It also has a higher amount of cholesterol than the chylomicrons had, pretty high proportion of phospholipids, and then some proteins as well. As we get to the LDLs, the density increases. We've got more cholesterol. We definitely significantly more protein in comparison. And then the HDLs have the highest density. Look at all that protein, lots of protein there, some phospholipids, cholesterol, because they're picking up cholesterol and bringing it back to the liver. They're not depositing cholesterol in those peripheral tissues, but they're the cleanup crew. They're taking that cholesterol back for recycling. You can also look at the various different apolipoproteins that are involved and which of the lipoproteins they belong on. So ApoA1, ApoB100, ApoB48, ApoC, ApoE. And you can also see that it denotes whether there's triglycerides or cholesterol esters and kind of the relative proportion there according to the colors. So picture's worth 1,000 words. Go ahead and noodle on that one for a little while. Take a look at it and see if that helps better understand the process, better explain the process. As I mentioned before, we don't need to eat cholesterol. We make our own. What do we make it out of? You may have wondered. Well, we make it out of these 2 carbon chunks called acetyl-CoA. Maybe you've heard of the Krebs cycle. Maybe you're familiar with acetyl-CoA as one of the intermediates in the various stages of metabolism. So we're able to break down carbohydrates, we're able to break down proteins and amino acids, and we're also able to break down fats and fatty acids into these 2 carbon chunks called acetyl-CoA. So all of the macronutrients that we eat are able to eventually be broken down into that 2 carbon chunk called acetyl-CoA. The acetyl group is the 2 carbon part, and the CoA, coenzyme A, is this cool attachment on there molecularly. We have an enzyme in the liver called HMG-CoA reductase, and what HMG-CoA reductase is able to do is take a bunch of acetyl-CoAs and stick them together to turn them into these cool ring structures, such as cholesterol. So even if you eat a purely vegan diet, even if you don't consume any cholesterol, no need to fear, your body can make plenty. Other things that we want to keep in mind is that sometimes we don't have enough glucose for the brain, and the brain really likes glucose. It primarily uses glucose as fuel. The body, the liver can actually use acetyl-CoA. Most of that is coming from fatty acids just by nature of how many carbons are in that chain. So it'll mostly use fatty acids to create the acetyl-CoA that's used to synthesize compounds called ketone bodies. So the process is called ketogenesis, and in times of carbohydrate deficiency, the body is able to make ketones. Sometimes this happens in diabetic ketoacidosis. It can happen in pure starvation, and it can also happen in diets that are deficient in carbohydrate. So there are 3 different kinds of ketone bodies that can be created: acetone, acetoacetic acid, and beta-hydroxybutyric acid. The one that you typically see in lab values for diabetic ketoacidosis is going to be beta-hydroxybutyric acid in the hospital setting. That's what is most commonly measured. And there are some kind of symptoms of that. There is what's called kind of ketone breath. It's kind of this sweet, fruity smelling breath that is produced because there is a high amount of ketone in the body. So making those ketones in the liver, now those ketone bodies are released into circulation, and ketones are able to cross the blood-brain barrier. So they are able to provide energy to the brain. So in times of severe deficiency of carbohydrate, the brain will have a backup plan of a way that it can get enough energy to kind of keep going. So it's able to put those, it's able to take those ketones, turn them back into acetyl-CoA, put it in the place during the citric acid or Krebs cycle where we would use acetyl-CoA, and keep that circle turning so that we can produce ATP, produce energy. And later on, we'll talk about the various different electron transporters, NAD, for example, NADH, that we're creating and then putting through the electron transport chain. So either way, the end result is that you're getting energy, you're getting ATP from that. If we have too much ketone being produced, then we can actually, since ketones can be acidic, we can actually have ketosis or ketoacidosis. If the blood becomes too acidic, then the various enzymes of the body can't work, and you can actually die from acidity, from too high of acid in the blood or too low of a pH. So the blood's pH goes down as acidity goes up, because that's the way the pH scale works. The lower the number, the more acid there is. So mild ketoacidosis, there's some debate on, how harmful that is, if it's harmful, but severe ketoacidosis is absolutely lethal and something to be avoided. This does beg the question of, things like ketogenic diets and that sort of thing. It's nuanced. There are definitely a lot of different pieces that need to be looked at, but primarily people on a ketogenic diet are not going into ketoacidosis per se. They're not creating such a low pH in their blood that they are going to have, severely lethal effects. The concern with things like a ketogenic diet would be more along the lines of how sustainable is that long term, and also, are they maximizing animal-based foods and minimizing plant-based foods, and what are the health implications of that? So how is that going to affect the gut microbiota? How is that going to affect things like HDL and LDL cholesterol? How's that going to affect the deposition of cholesterol within the arteries and the likelihood of cardiovascular disease? So those are kind of the main concerns, would be more on a long-term scale of chronic disease prevention. So